We are preparing all.
Okay.
It starts.
Started,
Mhm.
but I could not see the other four speakers. Please get them onto the other three.
You can start. Okay.
No, because I want to introduce them so that if all the four are here, all the three
As,
are here.
as you tell the names, they are going to appear.
Okay. Ah, a very good afternoon to all of you. So, welcome to this panel discussion on environmental dosimetry. Uh, I'm Nagabhushana. I'm working as a visiting professor in the Professor Sonia Tachumi's group. So, first, I would like to invite Professor Sen Gupta, D. Sen Gupta from Indian Institute of Technology, uh, who is an expert in the environmental dosimetry. He has already done a good quantum of work and he has given an excellent contribution in the field of dating. I welcome Professor Sen Gupta for this panel discussion.
Uh, so, is Professor Tatumi going to ask us the questions, or how do we proceed?
Yeah. I will invite Professor Sonia Tachumi, who is the chair of this conference uh, and who is an expert in the field of dosimetry as well as the dating. So I invite Professor Sonia to take part in this discussion. And I also invite Dr. Polymeris from the uh, Democratis in the Athens from the Greece. I will also invite Professor Polymeris for this panel discussion. I invite now to take part in the discussion. Let me go through the questions that we want to ask. Professor Sengupta will be and Professor Polymeris as well as Professor Sonia will first. Professor Sengupta will give his comments for the questions that we have discussed, supposed to discuss, and then followed by Polymeris and Professor Sonia. So I have the first question here for Professor Polymeris. How to survey the accuracy and precision for the environmental dosimetry using thermoluminescence dosimeters? Professor Gupta.
Uh, I think the TLD dosimetry. I would think is an intermediate stage, not in infancy and not at the plateau level. So basically, I would think that more and more sensitive and efficient TLD material will be discovered in the very near future. And the efficiency for that will be greatly enhanced, partly because of various space missions, because now Mars and Moon and maybe Mercury, these are the areas. And radiation dosimetry is going to play a very, very important role in also the space travel, for touristic purposes. So now nanophosphors is one of the new areas, and I believe that accuracy and precision, uh, we have to demarcate into couple of thresholds. For example, low energy, intermittent energy, and high energy alpha beta particles as well as gamma radiation. What we want to do, and again indoor and outdoor. That also we have to quantify before undertaking dosimetry. Now, for a country like India, we integrate few years of data based on seasons. 1 year of data for the 4 seasons may not be the real reliable data representative of these things. So, taking into granted that no major interaction occurs, I feel that one should, repeatability of the particular type of dosimeters, and then intercalibration with other equipments, field-based equipments. You have now various types of gamma ray detectors, semiconductor devices, which you can take to the field, and you can have very good GPS data, and you can make a grid, and you can lay out, and you can make iso radiation maps. So I think all these things have to be calibrated. And one of the major things I would like to stress on the fact is most of the things are done not in a teamwork. You should have a geologist, you should have a geophysicist, you should have a physicist or an applied physicist, and then maybe people from material sciences. And then this teamwork is going to be the sole thing which will work in near future. If we just go for diversity and unity, I don't think we will survive in radiation dosimetry, and especially medical purposes also, as I know now new type of rare earth materials are being used for MRIs and various things, synchrotron radiation, etc. So this was the first thing. I hope I am not taking too much of time. Now, how can I go to the next question, or Professor Polymerase will add up to this?
Yeah, I think Professor Polymerus, if you had to add
Uh,
a few comments on this, you're
You
welcome.
know, according to my opinion, uh, I am not the most appropriate to suggest, but I would like to suggest that maybe this is a. I would maybe we can do a common. I don't know if in using luminescence we refer only to thermoluminescence and OSL, or even to radio photoluminescence. What is your question exactly? Are we considering radio photoluminescence as a luminescence technique or not? Because we can still do cross-check and surveys using both techniques, or even OSL, TL, radio photofluorescence, and I think this will be much beneficial. Because
Thank you.
the most appropriate technique for low doses, especially if someone goes and visits Chernobyl, for example, it will definitely has to use radiophotoluminescence glass dosimeters. They are most appropriate. However, we can still collaborate and work with thermoluminescence as well.
I think the problem of the thermoluminescence it is because it's the light exposure, when you put in the site. So maybe RPL is nice, because you can use RPL dosimetry even in sunlight, it's no problem. Um,
Okay.
and
Thank you, madam. Thank you, Polymerus. Let's move to the next question. Uh, how to estimate the quantity of energy deposited into the different living organism due to its exposure to the radiation? What I mean to say here is the whatever the radiation it is get exposed by the human beings, terrestrial animals, aquatic animals, plants, even the organisms, etc. Right? So we know that we could be able to quantify the energy deposited into the human tissue, right? And we develop the dosimeters. Here the question is that how to estimate the quantity with respect to the whole environment is concerned. So, Professor Gupta.
uh, I think again this is a part of various subsets, because uh, if you talk about plants and these things, they have a different mechanism for hyperaccumulation. And I think we have tested some coastal plants which seem to hyperaccumulate very rapidly. And some of our colleagues in China are utilizing it for extraction of rare earth using this hyperaccumulation of plants. Means the rootlets, they trap some of the rare earths. Now, this is one mechanism. Then you incinerate it, and you can get it without, disturbing the ambient environment. Other processes are very, I don't think they are sustainable processes. Which the biggest deposit in China, in Mongolia, they are using it, because I have seen papers where environmental aspects people have talked about in the proximal cities, and it is very worrisome. Okay, so the thing is that hyperaccumulation plants and hyperaccumulation in various, say, other biota or human beings. For example, we talk about radiation dose or radiation equivalent man and these things. For people, means human beings, I think age-dependent factor is very important. For plants, I think since they are, small time span, so hyperaccumulation of particular processes, for example, creepers and tall plants and the way they take up hyperaccumulation process goes on, probably we can replicate in the lab also using hydroponics and etc. by treating plants in the laboratory and then see how much they have taken uptake of radioactivity, and especially uranium plus 6. Until unless you change the pH, I don't think thorium has sort of a hyperaccumulation. Potassium is entirely a different way of looking at things. So these are these things. Aquatic animals. I think this is a very important area. Why? Because the disappearance of various types of species because of radioactivity is telling that higher the people higher in the food chain, they are going to get severely affected. Forams, both shallow level forams and deep benthic forams. If they get affected, that means the other crustaceans surviving on them will disappear, and naturally this is going to end up higher in the pyramid. So I think these low-level extinctions in the coastal areas, benthic, and all these things, and the radiation damage patterns in these things is an indicator that human being is going to get severely affected in the long run, especially I would say beyond at the top of the ladder and at the younger stages. Okay, means between 0 to 5 years or the very young persons and higher levels. So one of the thing is that we have different age groups try to understand what is the food they eat. For example, plants which are growing below the ground in Ramsar, for example, how the population is staying, and all these things will matter a lot. Because as far as I know, I am a believer that asteroidal impact was the reason for KT extinction. And please remember, the meat-eating dinosaurs disappeared. One of the conjecture is the meat-eating dinosaurs disappeared because there was no plant-eating dinosaurs. They had perished before that. Okay, because plants was not there. The effect on chlorophyll was not there. So even in Chernobyl, if you see the plants, they've got fossilized because of radiation. Okay, so they're building the world's largest solar plant in Chernobyl. Okay, so plants, you see just the fossilized plants. You don't see any other thing in Chernobyl. So this is one of the major reasons that without time-dependent fossilization, the reason the chlorophyll disappears, and even if you see plants growing close to thermal power plants, you don't see chlorophyll content. It has decreased drastically. We have made some measurements. We are about to publish that also. So this was one of the things. So once the food chain at the lower end is disturbed, the human beings are going to get affected. It is for sure, whether we want or not. So the small plants, microbes and all those things, how they get affected, how the plants which are hyperaccumulating, they get affected. These are going to tell you how the human being in the long run, because of these, is going to get affected. For example, for some heavy metals, we know for sure, lead and etc., chromium and arsenic, we know for sure that they are affected. Carcinogenesis is quite common. Mercury, we had the peculiar problem close to Japan, the mercury poisoning, severe mercury poisoning. So I think radiation is one thing where we have to look into broader domain, and especially in the indoor environment, I would like to put more stress because in the pandemic, people have forgotten about the indoor environment. Thank you. I think that's what I want to say.
Professor Polymeris?
after after such a reply from Professor Sengupta, I think I don't have anything more to say, but to emphasize that from my point of view, all the dosimetric techniques that I have been studying so far in the past deal with mostly are focusing on man, okay, the man itself. However, we can still remember that there are a lot of biomarkers inside every organism, each organism. Even our blood can be used in that direction. Even the blood of animals could be used. However, I think there are still many techniques and many areas, topics, areas for research, but I'm not so familiar with. Unfortunately, I'm not.
Professor.
Yeah, for me, I never did this kind of measurement, but I think you can use GLG also, né? And try to evaluate the equivalent doses, né? But I think in the case of the accident dosimetry, né? Maybe you can use another equipment, né? Like a analyzing camera or another kind of technique. Yeah.
Yeah,
Professor Nagabhushan, just to add one point,
please.
one of the richest, best areas to study this are the high background radiation areas
Mm-hmm.
and the groundwater, the lakes, and the rivers around it, and the agriculture growing around that area. And we
Exactly.
have lots, including Brazil, including Ramsar, including India, including China, including Australia. So this HBRA, the proximal conditions of the plants and all these things can be studied very well because these are classical examples. You cannot, remove the HBRA from the nomenclature. And when we talk about HBRA, this is not because of technologically enhanced natural radiation. This is present because of the minerals, because of this thing. So it is ubiquitous. So I think uh, there are 7 or 8 very well-known HBRs. Ramsar is VHBRA, very high background radiation area. I think these are some of the hotspots. They are, some inter-laboratory or inter-country, like, inter-comparison sort of measurements are carried out for various laboratory measurements. I think if these sort of studies are carried, say, HBR in Brazil, HBR in Australia, China, if there's a group which works and comes out with something, with certain regulatory framework, I think that will be, I think, the ultimate goal, which any radiation expert or dosimetric expert will really love to work on. I feel that way. That is the last thing.
Yeah, that's an excellent field and an opportunity for
Yeah,
the researchers who can make a combination of this personal dosimetry as well as an environmental dosimetry and the effect of this radiation on the multiple organisms and the plants, right, to take up. So probably those who are interested, we can put a proposal for different funding agencies. Really is a potential. There is a potential to get the funding. I would like to thank you, Professor Gupta, for suggesting an excellent field. And I would like to add one more point here for the concern that we are discussing. It is something like this exclusive for the research. There are already people have developed the concept of dose coefficient to normalize the environmental as well as the human dosimetric calculations. Like that means while calculating this dose coefficient, we have certain assumptions. Like any entities which are represented by a simple volume whose composition and density which are homogeneous, and whatever the radioactive nuclide which is uniformly distributed within the entire volume of the source, which is external or internal, whatever it may be. So, right. So, by estimating this concept of dose coefficient, so we can normalize this radiation exposure to the different organisms, I believe. So, I hope with the collective conclusion from all these discussions, we have a very good point here to take up further level for this for the sake of the human being. So next, let's move on to the next question. Uh, Now, are optically stimulated luminescence dosimeters are well suitable for environmental dosimetry, or do we need to use both TLDs as well as OSLDs for this environmental dosimetry? Professor Gupta.
I think I would like that there should be a very good synergy or symbiotic relationship between TL and
Vediamo.
TL dosimetry as well as OSL.
Ναι.
And when I say TL dosimetry, I would like to say the recent, more efficient TL dosimetry in various forms is new type of phosphors. But OSL, because OSL alone, I don't think it tries to explain various things because you expect ages to increase as a function of depth. Suddenly, on the top part, you see some pedoturbation or certain other turbation, effect of cyclones, effect of wind, and etc. etc. Now, it is very difficult until by alone to explain based on some sedimentation rate, take some uniform sedimentation rate, take other radionuclides like beryllium 10, etc. etc. So I think a cohesive one between the radiation dosimetry over a long term, of course, means a year or 2 years, over spread over few months, integrated dose, compare means taken into account for the at least the dose rate calculation, then taking about the OSL because OSL is very sensitive in the sense way of transportation because it doesn't have a single zeroing effect. But see, such a sensitive effect, if the denominator we put a parameter which is not reasonably reliable. So what we are doing is decreasing the reliability of what we have done by hard work. So this is my opinion. Thank you.
Professor Polymer.
The main point is that we should remember why this, even though OSL was invented for dating, okay, why it was adopted very easily for the personal dosimetry issues, meaning because it's more practical. Okay, it's easier to erase the signal by just illuminate it. We don't have to heat it many times because imagine having a lithium fluoride, you have to make one measurement. So with OSL measurement, you just give one photon, one pulse, laser pulse, and then you just take the material and you irradiate and you just illuminate it. So it's totally empty. So the main idea is that luminescence OSL, as far as personal and environmental dosimetry is concerned, is considered to be more practical, not more accurate. Okay, I think this replies to your question.
Okay.
Not to mention that I am a thermoluminescence guy. I started my PhD from thermoluminescence, so I strongly support it.
Over to Sonia.
Yeah, the OSL is the measurement is so easy and quickly, and is cheaper than TL. And I think maybe the problem is the energetic dependency. In the case of aluminum, and there is this problem. So we can try to develop other phosphors. But yeah, you can use in a comparative measurement. Just one thing, comment, Professor Susanna, there is some comment from
Yeah.
in the chat. I don't know if you want to read.
Yeah, I can. Professor Susanna, welcome for your question. It is, we are using plants to evaluate accumulated dose on that. We are detecting cytogenic damage induced by low doses of radiation. For example, counting micronuclei produced on them. So this is the comment from Professor Susanna. Actually, she's adding the point for our discussion. So what she's saying is that they are using plants to evaluate the accumulated dose on that. So they are detecting the cytogenic damage induced by the low doses of radiation, right? So they count the micronucleides produced on them on the plants. So it's just an add-on point for our discussion. I hope that will help for the audience to
Uh-huh.
understand the previous question that we are discussing. That is about the different types of organisms will get the different types of damages, different types of effects due to the radioactive nuclide. Thank you, Professor Susana, for your useful comments. I think we'll move on to the next question.
No, no, Professor Agnish, can I just interrupt the last part?
Yeah. Please, please,
See,
please,
uh,
please.
see, when we talk about OSL, uh, you know, it is not the interpretation of ages in terms of chronology. It could mean the way it is transported from one part to another. So the mechanism of transportation, for example, in the lowest Paleosol sequence, it could mean the climatic cycles, right? It could also mean from the rock surfaces how the material got eroded and then got bleached, photo bleached. So, dates per se means a chronology. I would think OSL is much more omnipotent, and the data means, you know, when once you have got a very good data, it could be interpreted for various applications, not only for simple chronology, quaternary dating, but
Mhm.
for understanding the way the particles, that is, wind-borne particles or the fluvial particles from the rock surfaces got removed and how they got transported and in what sort of a regime, say, Paleosol Loess sequences are there. So, you know, simple understanding of the ages will tell you a part of the story. But if you want to add up, say, the climate into the Paleosol lowest ages, lowest especially is done with both TL and OSL. I think all these other things are also needed to be considered because then the work becomes more valuable. You know, Uh, see, in terms of earth and environmental sciences or something like that, environment is also the ambient environment. I call it the paleo environment is very, very important. And especially when in neotectonics, when you talk about new tectonics, especially we are close to the Himalayas. So when you talk about neotectonics, so the tectonic lineament, how they formed, you will get some ages, but they're sensu stricto, they may not meet the chronology. They may mean that when this isotectonic lineament formed. So, age, maybe, people may consider it to be a simple age in terms of chronology, depth dependent phenomena or sedimentation dependent phenomena. But I feel that it has a wider connotation. It OSL ages, I would not say. The OSL data says a lot of things, and I think one has to go thoroughly and to understand and take out maximum from uh, the data results. One can go and think and toss and get more interpretations, implications, and these are very fascinating things nowadays. I've seen a lot of lowest data, ages data, when they have talked about the environment as an important component recently by the Chinese groups and other groups. Anyway, you can continue. Sorry to disturb.
No, no, it's a very good, valid point
Mhm.
for the discussion. So the next question, here it is. Sometimes we see negative doses in environmental dosimetry. So what is the reason and how to avoid or correct such negative doses in our results?
See, I would believe that one way is to look is it just intermittent? Means it is just one of few locations and at a particular time of a period when you saw these negative doses. And if it is over a large region and these things, then of course we have to think of suitable corrections. But if it is just at some specific intervals, not across the entire dosimetry, say spatial extent, or if you talk about subsurface vertical distribution, then of course I think why this occurred may be looked into or delved deeply into the reasons. If it is more frequent, if it is less frequent, then we have to think about it, how to correct for it. So it is almost like big data analytics. In geophysics nowadays, most of the things we are doing with data analytics, like machine learning, and so the better, larger the database, these errors, which are mostly because of the signal to noise, the noise creating a big problem in the signal, gets more closer to one or two, and this creates a lot of problems. So, just if you take a long-term data in terms function of time and spatial locations and all those things, or say surrounding an area in 2 pi geometry, then if we get it, then we have to look for the reasons why this is happening. And of course, then we have to use some complementary techniques to explain why this is happening. Okay, uh, because I would feel that the radionuclide as a contaminant should be uniform in all the directions, and you know it should not be localized at certain points. So when the dose happens like this, probably there's some reason why this is happening. One should look for that, and probably if it is happening more frequently at particular locations, then of course there should be certain reasons. For example, one of the classical examples which I will tell you, the radiation dose in some parts of Orissa, where we have working now in the western part, about 500 kilometers, some of the part, we saw very younger data as compared to the lower part. So it was not a succession of increasing ages. Now, this then we realized that is the way the marine uh, winds and the cyclones and other things, hurricanes and the tsunamis affected, especially in case of tsunamis. We have done some work on tsunamis and the TL rates, and we see that suddenly the succession changes abruptly. So naturally, this will be reflected not only in the ages but also in the radiation dose also, because the entire different material which belongs to the tsunamis, high tsunamis, this is there. For example, another small point I want to make: the Fukushima disaster was not because of seismicity. It is because of a tsunami which followed a seismicity, which created a 15-meter wave height. Now, this 15-meter wave height will naturally carry some fine-grained particles, right? So, your dose, radiation dose will obviously be affected. So, I don't believe that 15-meter high tsunami waves will not affect the coastal areas or any other thing. So, any such disturbances, I believe, will have certain suspended particles, and that should be reflected in the radiation dose. So, negative dose may be suddenly you get these effects when the entire thing sequence is reversed. Means it gets admixture of various particles which are not the really the belonging to that particular location. These are not in the surrounding particles, and this creates a lot of problems. That is my what I feel. I may be wrong. Okay, thank you.
To be honest, I have not. I'm not familiar. I have not come along again with such a situation with negative doses. However, if ever it comes to occur to me, the first thing I would consider is statistics, probably. However, I am not familiar with the situation, so unfortunately, I cannot help you more.
Professor
Uh,
Sonu?
um, one time on two sites, I saw not negative dot, but a very, very low content of radioisotope. Eh? It's because the radioisotope can go down in the sediment. Do we have rain? Um, then we determined low dose in this site, but negative dose I never see your thing. Maybe the problem just you have to increase the sensitivity of your equipment.
Yes, I
Mm.
fully agree with Professor Sonia. Sensitivity is one problem. Yes.
Professor Gupta, we have a question from one of the audience,
Hmm.
Dr. Rene. The question is negative dose an error of calculation, or it is from where we get this negative calculation? There is a meaning maybe. Is negative dose an error of calculation?
See, calculation again depends when you calculate it. It again depends on the uh, abundance of the radionuclides present, right? Now, if the sources from multiple sources which do not belong to that region affected, and in addition, as I said, sensitivity. Sensitivity means the signal to noise, right? That means the ambient levels which a dosimeter can measure, minimum thresholds, and close to that, you know, I will not be able to, deconvolute the signal if it is very, very close to the background, right? So the problem is that if very, very low, for example, if your region, you have less than a ppm of uranium, thorium, potassium, right? Now, if there is a dose rate, say, accumulated over some time, it will be very, very low. Now, how to deconvolute that particular thing efficiently from the background? I think that is a very challenging task, especially for low background areas where you have only few ppm or less than a ppm of parts per billion of uranium, thorium, potassium. Potassium, of course, is percentage. I'm not bothered about potassium, but thorium is uranium. And you obviously know that in some countries they have used depleted uranium for warfare. Okay, so uranium, the ratios are also not same. See, generally we take that uranium 238 is more abundant, uranium 235 is less abundant. Now, how do you know that that is not an extrinsic material where the ratio, isotopic ratio, is not 1 by 1338? I don't think so. A priori, you can understand that. So the isotopic ratio of that, thorium is 100%, we are not bothered about that. But the way this uranium-238 to uranium-235 behave in a particular environment, and of course, the fractionation, I'm again telling you, uranium plus 6 and uranium plus 4, classical example, how they fractionate. In particular, especially mobile condition is one is water, hydrothermal solution, any solution activity, it may have leached out. So how do you ensure that it was a closed system in that small area where you imagine you put your phosphor? I don't think you can extrapolate that data. This is my belief. So statistics and data analytics may help you, but for such low levels, you require certain specialized techniques, very efficient phosphors, or some other way to look into the deconvolution of the signal to the average background because of such low levels of uranium, thorium. Potassium is always percentage. I'm not bothered about percentage potassium.
Thank you, Professor Gupta. Professor Polymoris, do you want to add any final comment on this discussion for this environmental dosimetry?
Not comment, but I would like to ask Professor Sengupta, nanophosphorus, were indeed invented, or the concept of nanophosphorus was adopted in order to, because we need to solve some problems in luminescence dosimetry, or just because it was a trend of the 2000, of the age of our years, to work more in the nano-sized materials? Because I'm not so much I have no experience in working in the nanoscale. Because, for example, I remember Professor Kortov saying to me that, okay, aluminum oxide in the nano-sized scale could solve some among the main disadvantages of the aluminum oxide, for example, thermal quenching, but the luminescence efficiency was not even close to the one that the micron size material has. I think you can understand my question. Or
Yes, just to add to your statement, when we started, thermoluminescence was started way back, a long time back, 40 years back, by Professor Singhvi, and I was there and that institute doing PhD. Now, that time, the other laboratories were doing uranium, lead, and various other dating, long-term dating, on an ensemble of grains. Presently, the monoxides which I have picked from the coastal areas, I am doing single monoxide dating using uranium lead. Same with dosimetry. So, from an ensemble of quartz, feldspars, etc., we have come to the stage when we have to look at a set of grains to look at not only the OSL signal, at the dosimetric level also. So, the at initially the number of quartz grains in the alley quartz was very large. Now I don't think such large ensembles of quartz grains are required for dating or any other aspects. So, we are going to a level when, we are going to understand the region. For even, we have done measurements on a single grain of monazite where the rare earth changes. This is electron microprobe, we have used it, and we have supplemented it with other dosimetric this thing. When the rare earth like yttrium and other things, neodymium, changes from the rim to the core of the monazite, the dimension of the monazite was 100 microns. So this can be done. This is doable. We have done it, and we have published a paper also. So the dates we are trying now is single grain dating. Of course, that is for some other purpose, but for understanding the coastal changes in the berm region, we are using optically stimulated luminescence again on grains, which we know for sure that see, we can pick up the grains in completely light tight area, no photons and no external light, but we can also use subsequently we can see the grains under a high resolution microscope or even on the screen and tell you what is the nature of the quartz. For example, if the quartz are rounded, naturally there's more fluvial action or aeolian action. So once we have used the same sample for dating, we can see those grains once OSL has been observed in high magnification microscopes or EPMA or other similar equipment, and then we can come to a better judicious implication. I think that is what we are doing. Maybe we fail.
I would like to add one point for Professor Polymer's question, because as we know, the nanophosphors are not very suitable for low dose dosimetry. Because of this large surface to volume ratio in the nanoparticles, the concentration of defects which are created in the crystallite, they are extremely small, which is not favorable for the creation of defects. However,
exactly.
this itself is an advantage for the high dose dosimetry, like the nuclear accidents. In all those cases, we have the very high doses where the microcrystalline dosimeters will reach the saturation for high doses. So in such cases, this nanocrystalline dosimetry will be very helpful.
Okay.
I think that is a point that you are looking for, I believe.
Yeah. From my point of view, I am looking if I have one material. It was also the question that I made to a previous presentation before me. If I have one material, I'm looking. I will have a PhD student, and we will probably study it. We have, let's say, beryllium oxide, okay, and try to make it nano size, grinding or ball milling, whatever. As we remove, as we change the main the constituents, the grain size, the crystalline sizes. I want to see all changes. I want to realize what's happening. And this is my project for my next student. That's why I am asking. Because I don't want to see what's happening in the micron and in the nanoscale. I want to see what's happening as the crystallite becomes smaller and smaller and smaller gradually.
Yeah. Yeah, I understand. You are
Okay.
looking for the variation in the dosimetric properties
In
from
the
microcrystalline
dosimetry,
to nanocrystalline.
band gap, activation energies, everything, full width at half maximum, whatever can be quantified, we will try to quantify, and then we will try to simulate.
Yeah, that's a good task. Of course, the ball milling method may be the one good approach to go from the microcrystalline to nanocrystalline. At the same time, you can also look for some bottom-up approach methods where you can start with the nanoparticles where Bohr exciton radius matters for the
Uh-huh.
properties. Then you keep on increasing the crystallite size or the nanoparticle size, right? Then certainly you will find some barrier at some certain size, at certain exciton Bohr radius. You will see the change in the properties, certainly,
Okay.
right? So maybe the both the ways. Of
Uh,
course, this is not an easy task. It will take a long years and many people to
Okay.
get involved, right? So of course, it's a
uh,
really good
uh,
task, right?
Professor Nagabhushan, other small
Sir,
thing, maybe
yeah,
the nature of rare earths, which you dope, use as a doping agent, may also help in looking at the sensitivity, different types of, rather than normal dysprosium, etc., etc. You can
yeah,
get rare earths, specialized rare earths, with which you can dope at a particular level, right? And then you can
yes,
decide on the optimum level, and then
yeah,
you can look at to this. Now, just to give
right,
you a simple example, we have done a lot of measurements on the grain size variation of the domains where thorium, that thorium in the beach, the peak is 200 microns. That is the peak of the grains, right? But the domains of thorium is present between 35 to 50 microns. But again, this is restricted to our 500 kilometers. I don't want to extrapolate to the rest of the world. Please, 6374 kilometers is a large area. But in our samples, the thorium rich domains in monazite are between 35 to 50 microns. And this has been done by high purity germanium detector, very ultra sensitive detectors. And so, you know, since this is true for natural samples, natural monazites, I think the phosphors can mimic some of these, and then you know we can see the grain size variation as well as a particular role of rare earths, where the depth from the conduction band and the role in the forbidden gap. Which type of rare earth is good for which type of substrate? That can decide, and how much you add can decide on this. So I think the rare earth story is going to go for a long time. I'm sure about it because I'm telling you, I've been working for the last 30 years. When I did the first dating in Sri Lanka, we were the first to date the coastal areas in Sri Lanka with Singvi and Deranegala in the EPSL. The story goes on and on. It is becoming more and more instructive. And 45 years hence, I stand here and I'm telling you the rare story goes on. So probably, we have to look at small scale, uh, nanoscale, nano dimensions, and then look at the nanophosphors and the role of the rare earths in a much, uh, you can say, much incisive manner, and then see how this grinding and all these things occur. Because I'm telling you, very soon you will get nanophosphors, which are quite different from what the nanophosphors we know today. And this is not what I am trying to talk like, uh, you know, uh, sort of astrologer or something like that. I'm telling you this is true because it happens in natural crystals. I myself seen in fluorides and various types of things, small addition and the color changes. And I've seen this in my field quartz and various things. A small addition across the grain boundaries, the entire quartz crystal becomes opaque. It is amorphous. There is no more crystalline. But a small amount. It's not 100 ppm. A small amount. Only the age is a parameter which is very important. Of course, it can be mimicked with pressure and temperature also. You can go down and this thing can be hastened up. So, I think nature is the biggest lab, and we can see certain materials uh, which are really important, rare earths I'm talking about, and they can be added to the substrate, and you can make it more and more efficient. Try to understand how the holes and the electrons they come, and in what is the response of the earth. Is it only a single high temperature glow curve which we are getting, which is very stable? All those things have to be seen. I'm not telling, but my gut feeling is the story of rare earths has just started in the nano dosimetry part. This is my gut feeling.
Okay.
I would like to add a point for Professor Sengupta, whatever he has mentioned about the using the rare earth ions for the nanophosphors. We have done a work on this same aluminum oxide in the nanocrystalline structures. We have added and we have optimized with the uh, uh, TB, which is terbium, and
Uh-huh.
thulium, TBTM, with the codopent, and we have optimized the sensitivity also for this ion beam dosimeters. So we took this task and we are doing this work, and just another few characterizations need to be done, like the fading and the other characterizations, right?
Correct.
So the part of this work is already been published, right? So maybe the same work can be extended for this environmental dosimetry also. Right now, we are concentrating only on the personal dosimetry, which can be used for the cancer treatment. Right? Maybe it can
Uh-huh.
be extended. Right? So, as Professor Gupta has already mentioned, there are different groups across the world in the different laboratories working on this doping rare earth ions into the nanocrystallines to increase the sensitivity, to decrease the fading, to get the proper linearity. Of course, there are many factors comes into picture the moment when you're talking about the different rare earth ions into the host matrix. Right? So Professor Polymeris is a very good input for all of us. So we can think of in Professor Gupta's concern. So maybe since we are already crossed the time limit given by the organizers, I would like to finally
Can I,
add one point from the one of the listener again, Professor Susana. She has made some comments. Different materials used in the luminescence dating have distinct characteristics and are commonly found together in natural samples that need to be separated, particularly with respect to the uh, dunes or the sand or the maybe the sediments that she is talking about is the separation of the quartz and feldspar. It is not a very simple task that she is saying.
right?
So by evaluating the literature, there is no agreement on the issue of the sample separation among the different researchers. So of course, if you could be able to establish one effective method to separate these quartz from the feldspar or removing one from the another, that may be an excellent explanation for many problems that we are encountering in dating. So there is one comment that has suggested by Professor Susana. I hope all of us agree with this point.
Yes. Another small point, I would like to add for our next decade work. I don't know. I'm a little bit on the other side of the rope. I'm talking about the younger people like Professor Nagabhushnam and Professor Polymeris. Even Professor Tatumi is younger to me. So what I am telling is that we have to take, like Professor A.S. Pascoa, Anselmo S. Pascoa told, once he wrote to me, we have to take technologically enhanced radiation far more seriously rather than taking it in a very lackadaisical manner, utilizing everything from plastic bags to melting everything and utilizing it for common use rather than thinking about what is going to be the effect on the human beings. And this use, reuse, and multiple uses of various steel, iron, other materials, from waste products. it has to be seriously thought about. We have strong groups working on various other contaminants, arsenic, fluoride, carbon dioxide, sulfur dioxide. But I think in terms of radiation dosimetry, uh, we should all get united together to say technologically enhanced radiation, natural radiation, is one of the major issues which will face us in the coming decade. I think if we focus on this aspect and then try to quantify those effects and the entire thing, then we can talk about remediation. Because remediation, whether we use anything, any human being, I don't think it doesn't create a problem. So remediation is, utilization is one part, remediation is the ultimate goal. And I think it is high time that we talk about remediation due to technologically enhanced natural radiation. Just shielding ourselves in islands, lead shields and various other type of shielding material, and saying that the world around, I'm not bothered. It is not the right solution since the population growth and the requirement for water and all these things are increasing day by day. So I feel TNR should be projected as the ultimate thing for anybody who works on radiation dosimetry. That is my belief, and that is what I would, the fag end, I would like to suggest to the younger colleagues who are working in this area. Okay, thank you. Muito
We thank
obrigado.
you.
So thank you very much. I think we can conclude now. So, madam, you want to add any final comments?
Just one comment to polymers. If you want to see the crystal size increase or decrease, you can change the temperature of treatment. If you are centralizing some phosphorus, if you increase the temperature of treatment, heat treatment, the size and usually the size of the crystal will increase. And so you can obtain crystal with different size. And
Okay.
to conclude the session, my speak. Yeah, I think Professor Sengupta is. I am in agreement with him. Now you have big issues, ambiental issues to solve. No? It is increasing, the pollution is increasing in high rate. So, and the radioactivity problem also now with this accident problem. So I think the young research will have a lot of work to do it. It's okay. So it is all.
Thank you, madam. I would like to summarize the concept that we have discussed. We started with discussing about the accuracy and precision of the environmental dosimetry by TLDs, and followed by the estimation of the quantity of energy deposited on the different living organisms, and to estimate this energy deposited, maybe using the concept of dose coefficient. And also we have looked into the concept of different OSLDs, whether they are suitable for environmental dosimetry or not, and as suggested by Professor Sengupta. So along with along with OSLDs, is always advisable to use TLDs in the dosimetry. And we have also discussed about this negative doses due to this difference in the background radiation and the estimation by the dosimeters, the dose estimated by the dosimeters. And we have also discussed a few concerns about these nanocrystallites in the dosimetry, in the environmental dosimetry for different sizes. And finally, I would like to thank all the speakers, particularly Professor Sengupta, who has given his valuable inputs, and his presence added an excellent value for this session. So thank you, Professor Sengupta, on behalf of the organizers and on behalf of the institution for your valuable time and valuable presence. And I
I'm
will
also
also
equally thankful. Yes, please go ahead. Go ahead. Then I'll talk.
Uh, I would also like to thank Professor Polymeris for joining us. His questions are very useful for the young generation, those who want to take up the research. So again, I would like to thank Professor
Not so
Polymeris
young, not so young still, but okay. Thank you very much.
from the organizers, from the institution, and from the all the Brazilians. And so we would like to finally congratulate Professor Sonia Chatumi for given an opportunity to conduct this panel discussion in this is a different for the first time in this SDMF year series. We are having this kind of session in the previous two sessions. We could not have this panel discussion, and Professor Sonia given us an opportunity to take up this environmental dosimetry issue as a part of this SDMF event. So, from all the young researchers from our team, so we would like to thank Professor Sonia. So, I would
Thank
like to
you.
thank
Thank you.
all the speakers
And
and all the listeners. Right? If you want to add any final comments, you are welcome.
Professor Nagabhushan, I will add to what the comment was made. Actually, uh, it is the brain which should be young. I don't believe in the outer parts. If the brain is young, it is fascinating, and it's a neural network in the human brain which controls everything. You know, the people have done fantastic work when the brains were partially, not working to the full extent.
Decaying.
There are a lot of people. So I think people, if you are brain dead, you're gone. Your heart doesn't matter to that extent. It is the brain. And I think I will urge all the young people, well, like Georgios Polymeris, Nagabhushna, and of course Professor Sonia, because the brain is the major person after Almighty, which makes us do our destiny. Okay, thank you. Thank you once
Yeah,
again.
So,
Professor Stephen Hawking is the best example, right?
Yes,
So as we
yes.
know, right, he kept his brain active, right? Though his whole body was almost dead, right? He has given such an excellent contribution for this mankind. So not only for the physics, the whole mankind, he has given an excellent contribution, right?
And there are a
So
lot of
finally,
people here also in India who are,
yeah,
In
despite
India,
being
yeah.
handicapped, they are contributing. They have various problems, TB and other things. They're one of the best mathematicians and one of the best physicists I know of who are like that. So it is the brain which does wonders.
correct,
I hope you all agree. And
exactly.
I wish you a fantastic next decadal work.
Thank you very much, Professor.
yes.
Thank you.
So finally, I would like to conclude his idea is very simple. Keep your mind active. You will be young forever,
Yes.
right?
Yes.
So thank
Okay.
you very much. Thank you
So,
all.